next speaker today is Ralf Jungmann from the Max Planck Institute of Biochemistry. He'll be talking about his Allen Distinguished Investigator project with Jan Ellenberg at EMBL, and they'll be talking about systematic mapping of epigenetic marks to the 3D architecture of the human genome in single cells. Ralf.
So just let me say how nice it is to be back actually in Boston, and happy to see so many familiar faces actually from my postdoc time here, which I spent across the street at the Wyss Institute for Biologically Inspired Engineering. So I should bring along greetings from Jan, who is sort of with us here on the picture, so he cannot be here today. And as Katie said, what we actually want to do with this award is figuring out a way how to paint the epigenome. And so it's very good that actually I'm after phase, so I don't need to do a lot of introduction about what we're actually after to do. So very similarly to phase approach, we don't want to use any sort of grinding up of the chromosomes and then do downstream sequencing, but we actually want to use a fluorescence microscope to visualize it in situ in a single cell. And so you could argue it's a type of an omics approach, and a former postdoc of my lab termed this localizomics. So it's a combination of localization microscopy like STORM or PALM, for example, with an omics type readout where you look at hundreds of molecular components or species at the same time. And how we're going to achieve that is we're going to use what you could argue is dynamic DNA nanotechnology. So it's always going to be probes that are labeled with a fluorophore but are based on DNA interactions that you can program on a computer more or less. So that also gives you a little bit of an overview what my lab is actually concerned about, mainly, is a little bit of instrumentation, which I'm not going to talk about today, but today I'm going to tell you what we actually need to achieve this, to paint the epigenome with single protein or single molecule resolution. So you need molecular resolution. We can have the argument what that actually means sort of on a biological scale. As I said before, we want to look at hundreds or thousands of molecular species, so you need some way of clever barcoding these. And during the course of the project that we did together with Jan now, we realized that nothing of this will have any influence if we cannot properly label stuff inside cells. So actually a large group of my lab is now become a biochemistry lab. So I'm going to touch a little bit on that. But so first, how are we actually going to do that? So let's say you would actually want to visualize a single gene localizations, 1,000 base pairs or something like this in a nucleus of an intact cell, together with the epigenetic modification of it, being it histone modification or methylation to DNA. There the problem comes in if you use a fluorescence microscope, is that's what you see if you look at a single molecule, right? So the pixel size here is about 100 nanometers. So hands up in the audience who knows how super resolution microscopy works. That's a few hands. Other tests, who does not know how super resolution microscopy works? There are a few hands. Okay, without going into too much detail, let's make a little bit of an interactive experiment. Let's say the size of this room is the size of what's called a point spread function of a microscope. So usually it's 200 nanometers in X, Y, and 500 nanometers in Z, but now we scale it up to the meter scale. So what I want you to do is basically tell me your first name when I count down from 3 to 0. So let's do that now. 3, 2, 1, now. Yeah. Okay, so I cannot demultiplex this information, this audio information that comes from you guys, because you're like fluorophores in the cell nucleus, illuminated with a laser at the same time, and you answer back at the same time. So there's no way to tell you apart. And what super resolution microscopy does, specifically stochastic super resolution microscopy, is shut you off most of the time, and then only every one and so often one person shouts their name out in the audience, and then I can pinpoint and localize it, and I do that a couple of thousand times and then reconstruct a super resolved map of this auditorium. So let's assume further you guys are proteins, and the spacing between your seats is about 5 nanometers. So the whole audience room here is then about 200 by 400 nanometers. So it's a diffraction limited size, right? And so at the end of the day, we actually thought with our technique that's actually called DNA paint, which uses transient DNA hybridization interactions to make molecules switch between bright and dark states so you can super localize them downstream. How far can you push this technology? And it's actually nice to have Minje in the audience here, who's a PhD student I worked with during my time here at the Wyss. And so we sort of pushed this technology and thought, well, how far can you actually push it? Can you get molecular resolution? And so what this technique actually allows you is to transform this image, the fraction limited, to actually resolve a structure like this, and to actually sort of zoom in a little bit more. It's a synthetic self-assembled DNA origami structure where you have points spaced 5 nanometers apart. So those are DNA molecules that are self-assembled on a little grid with a spacing of 5 nanometers, and what we do is we image them with a transient complementary strand that binds to them, and we super localize it. You can also do that with another logo for, in my case, obvious funding reasons. So it's the logo of my university that I'm affiliated with. So what's important here, if you measure the localization precision, and this is not something that's calculated from the localization precision of your algorithm or something like this, is really measuring the spread of these single sites, you get about a nanometer, okay? And that's the size of a dye molecule. So your localization precision is more or less at the edge how good it can get. And maybe we can have a coffee discussion if you can actually get better than that at the end. But what does that practically mean? Do you now have the technology at hand to actually transform that into a cell? So what you usually do to look at DNA molecules in a cell, you do DNA fluorescence in situ hybridization of fish. And so here, a postdoc in the lab, Mahipal, has actually applied this to telomere imaging, so nothing new here, basically end of chromosomes. You do fluorescence in situ hybridization, and then you can actually super resolve telomeres in cells. You also can do that on epigenetic marks. If you put the DNA strand on an antibody, and this is a secondary antibody now, and you label histone modification like this H3K9AC, it's an active mark, with a primary antibody, then you get basically this diffraction limited, and that's a super resolution image. So that's very nice. You can also do that with a repressive mark, diffraction limited, super resolution. So that's great. So you could argue, well, what's the problem? You've solved it, right? So you can sort of do that, and but not so quick, right? So if you look at normal protein imaging like microtubules in a single cell with primary and secondary antibody staining, and that's what we've done also with the histone modifications here. You can color code them in 3D. You see blue is sort of microtubules that are very close to the cover slip. Red is sort of further down in the cell. And you zoom into one of those areas, and I said to Alex and Tommy, who took that picture, well, can you make an XZ cut through these 2 microtubules that you see here, right? And so when you do that, when I first saw the image, I was completely blown away, and I said to them, well, you're looking at the hollow structure of microtubules, right, with a fluorescence microscope. Nobody has ever seen that before. And then you go in and measure the diameter. So cell biology 101, the diameter of a microtubule is 25 nanometers, okay? If you measure this, it's 57 nanometers. So that's not good. So either your microscope is not calibrated correctly or something else is wrong. But if you think about it, the localization precision of the technique is so good that you're localizing probes and not proteins anymore. So you're localizing the DNA strand on the antibody or the secondary antibody and not the tubulin protein. So that was more or less the turning point where a little team in the group, Thomas, Max, and Florian, set out to say, well, we need better text for protein labeling if we're ever going to make this work in a cell nucleus and co-localize sort of gene locations with epigenetic modifications downstream. So what tags for protein labels are there? So I said before, primary antibodies, if you use primary secondary, you actually double the size roughly. Then there's smaller tags like genetically encoded GFP molecules with a GFP nanobody. So there's the GFP and a GFP nanobody. Then there are other genetically encoded tags like halo tag or snap tag, which are sort of enzymatic tags that have ligands that then do a sort of covalent link to each other where you can put a dye on. And there's a little bit of a mystery molecule here that I don't want to reveal quite yet. So we turned our attention first to genetically encoded tags like the halo and the snap tag, and that's actually was a fantastic collaboration with Jan on this occasion, obviously, because he has all these genetically engineered cell lines where you have a homozygous replacement of proteins that all have a halo or all have a snap tag. And for this, we turned our attention to the nuclear pore complex. So you've seen a little bit of that in Cloda's talk yesterday. So the nuclear pore complex has these sort of double ring arrangement that's depicted here on the nuclear side of the nuclear envelope. So the nuclear envelope would actually sit here, and there's the cytoplasmic side of the nuclear pore complex. And what we're looking at structural proteins NUP107 and NUP96, which are part of the so-called Y complex in the nuclear pore. And so you have 2 copies in these symmetry centers here on top and the bottom of the ring. And so in total, you have actually 32 copies of these proteins. And we've labeled them N-terminally and C-terminally with these genetically encoded tags, halo or snap tag. So what does that allow us to do? So if you look at that in a fluorescence microscope now, here would be the diffraction limited image. Here's the super resolved image. So you see you can reconstruct this 8-fold symmetry of these nuclear pores in single cells, and then can actually compare, well, how good does a snap tag work compared to a GFP nanobody, compared to a snap tag on another protein, compared to a halo tag on the other protein, or compared to classical antibodies. And the analysis that we just did here is very simple. So it's a center of mass alignment and averaging of these structures. And what we then do is we measure the diameter or the radius of these rings, and also the width of these rings. So the width mainly corresponds to your localization, precision, or accuracy. So the smaller the probe becomes, the more defined the ring should be, and the diameter actually tells you if you're actually measuring the right distances. And what turns out is you can compare that with EM or cryo-EM maps, and the true value from these cryo-EM maps is the dotted line that you actually see here. And so for the SNAP and the HALO and the GFP nanobody here, you see that actually fits quite well the diameter. That's actually the true diameter of these pores. But the antibody, not only is the spread a lot larger, so you cannot localize it as well, but you actually measure a diameter that's smaller than the correct diameter. And that most likely comes from the place that these antibodies have a tendency to bind more to the center of the nuclear pore complex. Okay. So then we said, okay, let's use the halo tech, and let's figure out if you can actually use this technique to gain single protein localization, what you then actually would also use for targeting epigenetic modifications. And so the halo tech, with a little bit of optimization in the imaging, allowed us actually to resolve these doublets of nuclear proteins in the nuclear pore complex. So if you overlay a few of them, they're spaced 11.8 nanometers apart. And that we were able to do that because you can now, with this resolution, tell apart the nuclear side of the nuclear pore complex and the cytoplasmic side of the nuclear pore complex. So tech-wise, I would say that's a good thing to do, right? So you could use that to target epigenetic modifications in the nucleus as well. But does it really allow you to look at 5, 10, hundreds of protein species inside the nucleus? And the answer is no, because you cannot genetically engineer orthogonal tags for all of these components in the end. So you can never do something like localizomics with this. But another sort of binder might be able to actually do that for us. And you've also yesterday heard some talks about aptamers from SomaLogic. They're called SOMAmers. So those are DNA analogs of antibodies. So it's a DNA aptamer about 15 to 25 kilodaltons in size. And the trick is that they don't use natural DNA bases, but they're actually modified DNA bases. So they are hydrophobic residues or side chains on T bases that mimic protein-protein interactions. So you get a lot larger variety and thus better affinity and specificity than a standard sort of non-modified DNA and RNA aptamer. So for us, that's sort of a marriage made in heaven, right? It's a DNA molecule, easy to synthesize, easy to extend for DNA paint. And the cool thing is that company has 5,000 protein targets apparently in the shelf, and the goal of them is the whole proteome. So that would be ideal. So we sent out and tested a few of those. And so here's an aptamer or SOMAmer against the EGF receptor in cancer cell line. So that's an A431 cell line here. So it's actually 4 cells on a cover slip here. So if you zoom in once, that would be confocal or diffraction limited resolution. You zoom in twice, and that's what you get actually in terms of looking at single protein receptors with a normal microscope. So you don't see anything, right? So if you now combine this with DNA paint, you can turn that into an image where you actually see these single spots, right? And so you think, well, what are these single spots, right? So you can go in and then you can measure some distances, for example, up here. So these 2 spots are spaced 16 nanometers apart. You can actually calculate how good is actually the label in terms of localization precision in the end. So if you overlay a few tens of thousands of these molecules, you get a spread in localization of about 3 nanometer. So that's the size of the aptamer. So we are right at the limit what that probe can actually do. And then the question arises, are these single spots really single proteins, right? And so if you look at the kinetics of the blinking of these single spots, and if you would assume it's a unimodal distribution, a Gaussian, for example, then you know it's a single probe that's on there. And Sebastian did that, and you really see a single modal distribution of binding events to each of these spots. That tells you there's a single aptamer binding potentially to a single EGF receptor on the surface. So if you compare that with antibodies, here's the SOMAmer distribution. This is what antibodies look like. And the reason for that is there's a single DNA strand here per protein, and here are multiple modifications on the secondary, multiple secondaries bind to a primary, so that's anything else than quantitative, right? All right, so what you can actually then do downstream, and that's actually a slide that I put in for Michael Elowitz. We talked about that the other day. You can look at sort of the EGFR receptor organization on these cell surfaces. So for example, what happens if you stimulate the cell with EGF, so it's the natural ligand of EGFR, you start to see these clusters. And if you look closer, you start to see these dimers. So this is more or less, to my knowledge, more or less the first time that you actually see homodimers of EGFR with a fluorescence microscope. So if you look at sort of nearest neighbor distances of these dimers, you see there's a 19 nanometer distance in here that sort of shows up when you do that analysis. Okay, so we would argue, well then, now you're there in terms of probe development for the protein side. So what else would you need in order to paint the epigenome? Well, you could use a technique that's actually been developed during my postdoc here at Harvard a couple of years ago called exchange paint, so that allows you to image sequentially multiple targets and so get away with the fact that you need different colors for different proteins. So here you would actually encode everything in the DNA sequence, you do the imaging sequentially, and you can do as many colors as you have orthogonal DNA sequences. So back in the day, we did 10 colors with this in 10 rounds. So that would be a way to actually look at multiple proteins, multiple DNA sequences in the genome. But then at the very end, we want to detect proximity between these probes, right? We want to figure out which gene is actually epigenetically modified, and we only want to light up the gene that has this epigenetic modification. And so also yesterday in the talks, we heard about proximity ligation assays and how that helps you to sort of reconstruct distances and proximity. So we want to implement something like this on the single molecule scale with super resolution capabilities. And it's something we call proximity paint. So, and this works pretty simply, actually, in the sense that, let's say you want to test the proximity of two target molecules here. So what we do is we split the paint docking site into two halves, where one half is labeled to one target protein, the other half to the other target protein. When the two are in proximity, you can form this sort of stem here, and then you get a signal. If only one half is present, you only have half a docking site, and because you're operating roughly at the melting temperature, your off rate is so high that you will not be able to detect this, right? And so Florian tried that out, and sort of on origami, there's the p-pen signal here in red, so it's spaced 20 nanometers apart. The cyan spots is just for reference here. And if you leave out one of the strands, you get no signal, right? And so this is actually an easy way to do this detection then downstream in cells. And so the last sort of 2.5 minutes or so, I want to spend on one thing that also came apparent. So you could argue, well, now you've solved all the technological challenges, right? And then we sat back, Jan and I, and did sort of a back on the envelope calculation, how long of an imaging sort of run we actually would need to reconstruct all positions of, let's say, 1,000 genes in a chromosome together with the epigenetic modifications, and not only in one cell, because you need some statistics, right? You need to do that in tens or hundreds of cells. And an astronomically large number came out of that. And so no PhD student in the sane state of their mind would ever do that experiment, right? So we thought, well, that there needs to be some more speed for DNA paint. And so the problem here is, if you look at this, is all basically based on DNA hybridization reactions. So that's rather slow, actually. So if you have an origami with a single site, you have 34 or 35 seconds between single binding events. Right, so if you change the buffer a little bit, you can up that. If you up the concentration of magnesium, you get a speed increase of a factor of 2. But now we turn to the sequence that we actually use for this, right? And so if you change a sequence from our old sequence called P1 and the new PS3, you can actually speed it up by a factor of 5. And you combine those together, you speed it up by an order of magnitude. So it's 10 times faster imaging. And what that actually is this is the old sequence and this is the new sequence. So without going into too much detail, we can talk about that in the coffee break if you wish. You basically strip the sequence off from all intermolecular interactions. So there are here T bases that can base pair transiently with A bases in the same sequence. So if you go to a 2-letter alphabet, TCC here, so you don't make any of these hairpin formations in the end. And that makes this sequence so much faster. It's an obvious thing if you think about it, but I thought with these short oligos that shouldn't make an influence, but it has a big influence. So now Sebastian in the group has actually developed another method that's actually now 50 times faster, so we now have a 50 times speed increase for these imaging modalities, and that should allow us to actually reasonably fast reconstruct these epigenetic modifications. So what does that mean practically? Here's a typical sort of DNA pane field of view, 100 by 100 microns, microtubules, 200 seconds imaging time. So now that allows us to tile these things at 144 tiles in an image acquisition time of only like 2 hours, which otherwise would have taken us a couple of days to actually get this. And I'm happy, last slide, to report that we actually adapted technology that's actually also used in the Allen Cell Explorer to actually have a Google sort of Earth-type view to browse this data. And so you can now go in and actually zoom at different zoom levels and get the molecular details without actually losing the spatial context. And you can play around with this at this website. All right, so that brings me to the end, not without thanking all the people who have actually done the work and you for your attention. I'm happy to take any questions.
We have time for one question. Or you can find Ralf over lunch. Thank you so much.
Thanks.